Hello, everyone, and welcome to the presentation that steps into our survey of animals. We're going to start with examining invertebrates, which we'll elaborate on what that means in just a moment. But in particular, we'll examine both the poriferans, also known as the animal sponges, and the cnidarians, sometimes called the stinging celled animals. Let's map out what our learning objectives are first. And this is actually really important for you because it will allow you to have a mental image of why we are going to talk about each topic and where it's all headed. Our first learning objective is to describe the organizational features of the simplest multicellular organisms. Then, you should be able to explain the various body forms and bodily functions of sponges. You'll be able to compare structural and organizational structures of the porifera and the cnidaria, and describe the progressive development of tissues and their relevance to animal complexity. Identify the two general body forms found in the cnidaria. Describe the identifying features of the major cnidarian classes. Animalia, like all multicellular life, has small beginnings. We can trace our very small branch on the taxonomic tree of life back to a common ancestor between early life and the choanoflagellates. These were protists. And you may recall from our protist lecture that protists are usually single-celled eukaryotes. This branch was called the opisthecons, owing their name to having a single flagella at the rear, much like the image at the bottom right. They tend to be unicellular, but interestingly enough, as you see in the upper left, there are also colonial forms. Colonies aren't multicellular organisms, but it's certainly a step in that direction. When you hear the words colonial organisms, think of a group of individual cells or groups of individuals sharing resources and coordinating their behavior. This would be contrasted to multicellular organisms, which are individuals made up of genetically identical cells, all originating from a single cell. In animals, this is typically the zygote. The protist choanoflagellates, again, they have that single flagellum at one end, and it's surrounded by a collar, just like the image at the bottom right. The collar has several microvilli. As the flagellum rotates, it funnels water and small bacteria in that water in toward the collar and microvilli that capture and funnel the prey downward for consumption. Fascinatingly, these types of collared and flagellated cells are produced by poriferans, and for the exact same reason for this sort of feeding behavior. These animal sponges are an early branching member of an animalia and a major topic of this presentation. Let's back up now and discuss this new term. We've never defined invertebrates, so what do we mean here? Invertebrates are animals that lack an endoskeleton. There are a few caveats to this, so we'll say it another way in that they don't have a vertebral column or backbone. Like many classifications in biology, this one has some exceptions, and we will encounter some of those later when we reach groups such as hagfish and tunicates. For example, even though echinoderms, shown in this image to the right with an iconic sea star, are invertebrates, they produce tiny calcareous elements called ossicles that are embedded in their dermis. These ossicles form a true endoskeleton covered by epidermis. So while they lack a backbone, they have evolved their own version of internal skeletal support, but they would still be considered a invertebrate. So for now, just remember that when we say invertebrates, we're talking about animals without a bony vertebral column. With that definition in place, let's begin our survey with the simplest invertebrates, the group called Parazoa. The name Parazoa literally means beside the animals, which tells you they sit right at the base of the animal tree. The phylum we are looking at is Porifera, the sponges. Sponges lack true tissues that are embryologically homologous to those found in all other animals. This is because they do not form a true gastrula during embryonic development. This is something we went over in our animal diversity presentation. However, they do have specialized cells that perform distinct functions in a way that looks and acts very similar to tissues. The important point is that these specialized cells are not derived from the same embryonic germ layers or developmental processes that produce tissues in more complex animals. This fundamental difference places sponges firmly in the Parazoa branch, separate from what scientists call the eumetazoans, which means well-developed animals. To put this evolutionary split in perspective, scientists estimate that the divergence between Parazoa and Eumetazoa occurred over 1 billion years ago. That makes sponges one of the oldest surviving animal lineages on Earth. Understanding sponges is important because they show us what the very first steps toward multicellular animal life looked like. You can see them here in this taxonomic tree at the very top. There's the Porifera, and they would exist outside of the Eumetazoa, which contains the rest of Animalia. So um, they are our link between the first protist choanoflagellates to multicellular animals. But before the evolution of the major innovations we discussed in the last presentation on animal diversity, such as true tissues, germ layers, and organized body plans. Let's look at some general characteristics of sponges before delving into more specifics. Sponges have a free swimming larval stage called either parenchyma or amphiblastula, depending on what species, and a sessile, that means a non-motile or it's non-swimming, in other words, adult stage where they attach themselves to a substratum as they feed and grow and reproduce. Their classic method of feeding, excreting waste, and gas exchange is through water movement. And they are masters at constructing canals, chambers, and cavities in which they stream water along and through for efficiency. This system ensures that the sponge stays in direct contact with moving water, making feeding, cellular respiration, and waste removal possible even in such a simple organism. Now let's examine how this water circulation works by looking at the basic body plan. Most sponges have a relatively simple overall shape that resembles a cylinder or a vase. Inside this body is a large central cavity called the spongicle. Water enters the spongicle through numerous small pores called ostia and exits through a larger opening at the top called the osculum. Although many sponges follow this basic tube-like design, there is actually quite a bit of diversity in sponge body forms. Different species vary in the size and shape of the spongicle and in how their feeding chambers are arranged. This variation reflects different solutions to the same challenge, moving water efficiently through the body so that every cell can obtain food and oxygen and release waste. Sponges may look like simple tubes, but inside they have a surprising variety of specialized cell types that work together to keep the animal alive. Even though these cells are not organized into true tissues, the way we see in more complex animals, Each type of cell has a very specific job. So, water first enters the sponge through the ostia. These are pores that are created either by specialized cells known as porocytes that line the ostia pore, or by folds in the body itself. The name Porifera actually means pore bearers, and these ostia are what give the entire phylum its name. The most important in defining cell type in every sponge is the collar cell or choanocyte. And whenever you see cyte in biology, what that means is we're talking about a type of cell. So you might be looking at the immune system of animals and see lymphocytes. And so cyte means that it's a cell, and lymph deals with the lymph system. And there you have it. And each choanocyte has a sticky collar made of microvilli wrapped around a single flagellum. When the flagellum beats, it pulls water through the collar, trapping food particles that are then moved down to the cell body and eaten by phagocytosis. This feeding system is almost identical to the one we saw in choanoflagellates in the last presentation, which is powerful evidence that sponges and those protists share a recent common ancestor. Choanocytes are so versatile that they can even change into sperm cells when the sponge reproduces sexually. The short video on the left shows a poriferan with a neon dye added to the water, and this enables you to visualize that flow of water out of the osculum. You can imagine incoming water entering through its ostia around its body wall and exposing the collar cells to the fresh water, allowing them to feed and then flowing out the top end. Beside the porocytes and the choanocytes, there's a few more specialized cells we want to cover. Pay special attention to the ones that make compounds, because the types and amounts of these compounds allow us to categorize sponges into three sponge classes. The pinacocytes form the outer protective layer, similar to how we have an epidermis, important for protection. Of the inner mesohyl, this is a jelly-like matrix between the outer layer and the feeding chambers. It contains collagen protein and carries several cells that are mobile within this matrix. Of these cells include the amoebocytes. You'll remember these because they are named for their amoeba-like movement, which they need to have because their job is to deliver nutrients to other cells and also differentiating into oocytes, which is a name for animal egg cells. Next, we have the sclerocytes. Just remember, sclerocytes for the skeletal materials. These skeletal materials are secreted by the sclerocytes to form the spicules, which are needle-like scaffolds. Together, these cells show how sponges have evolved ways to support their bodies and distribute resources even without true tissues. Lastly, we have the spongocytes that produce, no surprise, they make spongin, a protein matrix that is soft and elastic. You might say it is spongy, and this protein is found in the majority of sponges. Now we reach our first assessment question to see how you're tracking along with the material I presented. Checkpoint question 1 asks, what do sclerocytes produce in sponges? Most sponges contain small, bone-like structures called spicules scattered throughout the mesohyl. As we covered, these spicules are manufactured by the sclerocytes and can be made of either calcium carbonate or silica. Spicules serve two main purposes. First, they act as an internal skeleton that supports the sponge's body. and helps maintain its shape against the constant flow of water. Second, they can deter predators because many spicules are sharp or abrasive. As you can see from the image to the right and center, they are varied and don't look very palatable, if you ask me. The size, shape, and chemical composition of these spicules are so distinctive that biologists use them as a primary way to classify sponges into different groups. The presence and chemical makeup of spicules allow us to divide sponges into main classes. Calcarea has spicules made of calcium carbonate and contain no spongin at all. You can see an example of one of these in the upper left-hand side. The class Hexactinellida, often called glass sponges, has spicules made of silica and also lacks spongin. Some of these spicules can grow incredibly up to 3 meters in length. Class Demospongia is the largest group and usually contains a mixture of spongin fibers and silica spicules. The image to the far right is that of the bath sponge, Spongia officinalis. These sponges are the original natural sponges that people still use in everyday life. Checkpoint question 2. Which sponge class lacks spongin and has silica spicules? Now that we understand the skeletal framework, let's look at how sponges actually function on a day-to-day basis. Sponges have no complex digestive, respiratory, circulatory, or nervous systems. Instead, everything happens through direct contact with moving water and through a network of specialized cells that communicate just enough to keep the animal alive. Water enters through the ostia, passes over the choanocytes where food is trapped and eaten by phagocytosis, and then exits through the osculum. Small bacteria, anything smaller than about, well, less than 1 micron, let's say, are captured by the choanocytes. Larger particles that make it to the outer surface can be engulfed directly by the pinacocytes. Remember, those are the cells that make up the epidermis like surface of the sponge. Once food is inside a cell, amoebocytes act like delivery trucks, carrying digested nutrients through the mesohyl to cells that didn't get to feed directly. Another highly unusual trait for an animal is that all digestion in sponges is intracellular. There's no digestive tract whatsoever, so each cell must engulf and break down its own food particles. Because there are no specialized organs for gas exchange or waste removal, oxygen and carbon dioxide simply diffuse across cell surfaces, and nitrogenous waste diffuses out into the passing water. This entire system works because water is constantly flowing through every part of the sponge. The lack of complex systems is not a weakness here. It is perfectly matched to their sessile lifestyle and the way they let the environment do most of the work for them. Here's an interesting side note. Nothing you'll need to commit for an exam, but it shows how even these simple animals form complex ecological relationships. In some sponges, they host green algae or cyanobacteria inside their tissues. This partnership is called a holobiont. The sponge is the host, and the photosynthetic microbes are the photobionts. It works very much like the relationship between fungi and algae in a lichen. The algae, or cyanobacteria, provide sugar through photosynthesis, while the sponge offers protection and a steady supply of nutrients from its waste. In some cases, the microbes even live inside the sponge amoebocytes. This symbiosis can be so important that when environmental conditions change, the health of the entire sponge community depends on keeping these microbial partners happy. Another fun fact about sponges that won't be tested directly on an exam or a quiz is that while most sponges are filter feeders, evolution has produced some surprising exceptions. There are about 150 species of carnivorous sponges that have given up at filtering tiny bacteria and instead catch larger prey, mainly tiny crustaceans. These sponges use sticky threads or hooked spicules to snare passing animals. Once the prey is trapped, the cells grow around it, forming a temporary digestive membrane and releasing enzymes to break it down. The harp sponge, pictured here, is a dramatic example. Its branches look like a harp covered in Velcro, like hooks. This shows how, even in a group of ancient and simple or animals such as sponges, natural selection can produce completely new feeding strategies when the opportunity arises. Sponges are remarkably good at reproducing without sex, and this makes sense for sessile animals that can't go around finding new mates. There are two main asexual methods. In fragmentation, a piece of the sponge simply breaks off, often due to a wave action or an animal brushing up against it, and that fragment settles somewhere else and grows into a new sponge. In budding, a small outgrowth forms on the parent, and it develops its own ostia and osculum, and eventually either detaches or stays attached as part of a colony. Both methods allow a single sponge to quickly produce genetically identical copies of itself, which is an excellent strategy when conditions are favorable and the space is available. So far, we've been focused solely on sponges, but there also exist sponges that evolved to live in fresh water. We won't stop to explore their different members, but we will just take a moment to examine a particular structure they specifically evolved to deal with their unique environment. Freshwater sponges face a special challenge because the water they live in can freeze or dry up seasonally. To survive these harsh periods, they produce a special resistant structure called a gemule. A gemule is a tiny survival capsule. It has an inner layer packed with amoebocytes that can grow a new sponge and an outer protective layer that may be reinforced with spicules. When conditions become favorable again, when the water returns and temperature rises, the amoebocytes exit through the small opening at the top. It's called a micropyle. You don't need to commit that to memory, but it's called a micropyle. And they begin building a brand new sponge. Gemmules can remain dormant for long periods, survive drying out completely, and even tolerate freezing. This adaptation is one of the reasons freshwater sponges can recolonize habitats year after year despite living in an environment that dramatically changes with the seasons. This was a necessary adaptation because recall that sponges are sessile, so they can't just pick up and move to another watering hole. The freshwater sponge in this example here, the Spongilla lacustris, is a good example. You can see it here growing on a rock in a river. In late summer or fall, as water levels drop, it produces gemmules that wait out the winter or dry season and then hatch in the spring, quickly repopulating the area. Sponges can also reproduce sexually, and the process is surprisingly efficient for such simple animals. You'll recall that we said amoebocytes can differentiate into oocytes, and they'll stay in the spongocyte, while the choanocytes differentiate into the spermatozoa, which makes sense that they already are on the outside surface and they have a flagella, which makes sense that they become a sperm cell. Most species are monoecious, meaning a single individual can produce both eggs and sperm. The sperm are released into the water through the osculum and carried by currents to other sponges. When a sperm reaches an egg inside another sponge mesohyl, fertilization occurs, and the zygote begins developing into a larva that swims freely. That larva eventually swims out through the osculum and settles on a new surface and grows into an adult sponge. Some species even switch the order in which they produce eggs and sperm during the season to encourage fertilization between different individuals and increase genetic diversity. This combination of asexual and sexual reproduction gives sponges great flexibility in how they spread and adapt. And that's going to complete what we want to cover with the simplest multicellular animals, the poriferans. To the next major step in animal evolution, phylum Cnidaria. These are the jellyfish, corals, sea anemones, and their relatives, often called stinging-celled animals. The photo on the slide shows four very different members of the group: a large jellyfish, a gorgonian soft coral, a branching stony coral, and a colorful sea anemone. Even though they look nothing alike as adults, they all share the same fundamental body plan and the same innovations that separates them from sponges. In the previous presentation, we saw how animals progress from no true tissues in Parazoa to organized tissues in Eumetazoa. Cnidaria are the first phylum we encounter that belongs to the Eumetazoa branch. And today you will see exactly, well, in this lecture, whenever you watch it, you will see exactly what that leap looks like in living animals. Cnidarians are placed in the clade Eumetazoa, which means true animals. This clade is defined by two major evolutionary advances that sponges never achieved. First, cnidarians have organized tissues that develop from embryonic germ layers through the process of gastrulation. Second, they show the first clear division of labor among those tissues. Nearly all cnidarians, over 90% of species, live in marine environments. They possess radial symmetry, which allows them to sense and respond to the world equally from any direction. They are diploblastic, meaning they develop only two germ layers, and those layers give rise to specialized tissues. This combination of true layers and radial symmetry marks the first time in animal evolution that we see a body plan capable of coordinated movement and more sophisticated prey capture. Let's look more closely at what radial symmetry really means for these animals. In the diagram you see here, a sea anemone is shown from above. Any plane that passes through the central mouth and down through the body will cut the animal into two mirror images or halves. This arrangement lets the anemone or jellyfish interact with its environment from all directions. There's no distinct head or tail, no left or right side. For a sessile animal like a sea anemone or a drifting jellyfish, this is actually highly efficient. Food or danger can come from any angle, and the body is equally prepared to respond. We'll see later how this symmetry contrasts sharply with a bilateral symmetry. But first, another assessment question. Checkpoint 3 asks, which characteristic is true for Cnidaria? To understand why diploblastic organization matters, let's recall the embryonic process we covered in the first presentation on animals. During development, most animals separate their cells into germ layers. Diploblastic animals, like cnidarians, form only two layers, an outer ectoderm and an inner endoderm. Between them lies a non-living jelly-like layer called the mesoglia. There is no true mesoderm. This body plan is typical of animals that show radial, biradial, or rotational symmetry. Because there is no mesoderm, cnidarians cannot form the complex muscles, circulatory systems, or internal organs we see in the triploblastic animals later. Yet the two layers they do have are enough to create functional tissues that allow them to feed, move, and sense their surroundings far more effectively than sponges can. Arguably, the most important innovation in cnidarians is a unique cell type found nowhere else in the animal kingdom, the cnidocyte, or stinging cell. These cells are the reason the phylum is named Cnidaria. Each cnidocyte contains a specialized organelle called the nematocyst. Inside the nematocyst is a long coiled thread with barbs at the base. You can see the barbs right here. A tiny hair-like projection called the cnidocyl, seen here, sticks out from the cell surface before the thread is released. When the cnidocyl is touched, the nematocyst fires with incredible speed and force. The thread shoots out, entangling or piercing the prey or predator. Many species also inject powerful toxins through the thread. Some toxins are neurotoxins that paralyze the target. Others are pore-forming toxins that destroy cells. This stinging mechanism allows cnidarians to capture prey and defend themselves even though they have no jaws, no limbs, and often no ability to chase anything. It is one of the most effective weapons evolved in the animal kingdom. And here's the cnidocyte action. This image right here gives you an animation that represents what those threads would look like as they're shooting out from a tentacle. And remember, this is the primary way that cnidarians hunt and feed. The toxins can include neurotoxins that stop nerve signals or pore forming toxins. So what that means is the cell membrane no longer is fully intact. By opening those pores, then the membranes become leaky, and so that's cytotoxic. With an understanding of their method of feeding, their body plan will make much more sense to us now. Cnidarians exhibit two distinct body plans that represent a major evolutionary advance over the single body form we saw in sponges. The first is the polyp, which looks like a stalk or cylinder attached firmly to a substrate. The polyp is sessile, so it remains fixed in one place for its entire adult life. At the top of the polyp is a single opening that serves as both mouth and anus, surrounded by a ring of tentacles armed with stinging cells. The second body plan is the medusa, which looks like an umbrella-shaped bell. The medusa is motile and it drifts or swims through the water. Its mouth and tentacles hang downward from the underside of the bell. Many cnidarians are what we call polymorphic, meaning they display both body plans at different stages of their life cycle. In these species, the polyp stage is usually the asexual phase that grows and spreads by budding, while the medusa stage is the sexual phase that produces gametes. Importantly, both the polyp and the medusa are diploid organisms. And this alternation between the two body forms gives cnidarians great flexibility in how they feed, reproduce, and disperse. Cnidarians have really cool specialized polyps that are experts at doing certain tasks. Having specialized body forms within the same organism is called polymorphism. And we are going to use the cnidarian obelia as a classic example to show how this works. We'll go over the two major clades in a moment, but we might as well tell you that obelia is a member of the hydrozoan, Medusazoa. And many of the medusazoans are known for having both a polyp and medusa life stage. To accommodate the switch between polyp and medusa, they have two specialized polyp types. The first is called a gastrozooid. It's shown at the top of the image. Sometimes all of these names feel complicated to understand, but if you just focus on the root words, most of these you could probably write your own definition before even seeing them. With a name like gastrozoid, take a moment to guess what its job is. Well, see how it has its stinging nettles arising out of the top? Those will paralyze and capture their prey and funnel it down to their central mouth to be ingested in the what we call the gastrovascular cavity. In other words, this polyp's job is to capture and digest prey, where the gastro part comes from the gastrozoid. The gonozoids are the reproductive polyps. As you can see, they lack stinging tentacles because they aren't built for feeding. Instead, they produce tiny, free-swimming medusae by budding. These medusae break off from the gonozoid and swim away to carry out the sexual part of the life cycle. This division of labor inside one colony shows how these cnidarians can take a greater step in complexity while still remaining as a diploblastic animal. And this slide here shows that life cycle from colony to medusa and the sexual reproduction that follows it. The medusa produce gametes, and after those gametes are fertilized, you get what's called the planula larva. This larval stage is called planula because they're small and slightly flatten out, and they're a free swimming stage in the life cycle here. And eventually they will land on some sort of substrate and form a new polyp that will then go on to create a colony. Let's go ahead and look at this life cycle in a different organism, the Clytia. And so this first example here is showing the gastrozoid that's capable of feeding on prey. We can see the tentacles capture, stun the animals, and then bring them into the central mouth, the gastrovascular cavity, to be digested. And on the same colony, we have the gonozoids, and we can see them producing and releasing the medusa. Now remember, these medusae are now the ones that are going to be capable of doing sexual reproduction. All of this so far is just asexual. Now let's look at that. So here are some of those free-living medusa. They have some tentacles of their own, as well as some reproductive units as well. Let's take a closer look at that. So now they're making these spheres that are generating both sperm and eggs, and once fertilized, this will repeat here. Let's look at the repeat. So here we see the spheres of the gametes being produced. Once fertilized, they go through cleavage to make a blastula, and then this is our planula larvae. Which will find a substrate and then bud and develop into a new colony. And that is a common process that we're going to see in more cnidarians later on, especially the hydrozoas. But first, we can see how the body plans we just discussed are built from embryonic tissues. Cnidarians are, again, diploblastic, so they develop only two tissue layers. The outer layer is the epidermis, which comes from the embryonic ectoderm and forms the protective covering and the tentacles. The inner layer is called the gastrodermis, which comes from the embryonic endoderm and lines the digestive cavity. Sandwiched between these two living layers is a thick, non-living, jelly-like substance called the mesoglia. This mesoglia gives the medusa its buoyancy and flexibility, and it acts as a simple skeleton in the polyp. Within each of the two tissue layers, we find differentiated cell types that perform specific jobs. There are nerve cells for coordination, contractile epithelial cells that act like primitive muscles, enzyme secreting cells that begin digestion, and nutrient absorbing cells that take up food. These cells are connected to one another, allowing the animal to coordinate movement for feeding. However, because there is no third germ layer, cnidarians don't form complex organs or organ systems. The two layers and the mesoglia are enough for the level of complexity they need. Take a moment to look at the diagram on this slide. It shows a cross section through a typical polyp or medusa. You can clearly see the outer epidermis, the inner gastrodermis, and the thick jelly-like mesoglia in between. Notice the cnidocytes we just discussed are distributed in the tentacle area, layer. This simple sandwich-like construction is the foundation for everything cnidarians do. Cnidarians have a few more tricks up their sleeves that we did not see evolved in Porifera, but we're going to brush up on them just a little bit here. Cnidarians possess the simplest nervous system found in any animal. It consists of a diffuse network of nerve cells spread throughout the body. We call it the nerve net. Unlike more advanced animals, there's no centralized brain or spinal cord. In some species, particularly the swimming medusa forms, you may see slightly more organized regions such as branching nerve plexi or nerve cords, especially around the edge of the bell. These provide a little extra coordination for swimming and feeding. The individual nerve cells in cnidarians often combine both sensory and motor functions in the same cell. While this won't be covered on any assessment or quiz or anything like that, it's worth mentioning another evolutionary step related to their motor functions, and that's chemical. Instead of relying on the chemical transmitters you might be familiar with, such as dopamine or serotonin, many cnidarians use peptides, which are short protein sequences, as their main signaling molecule. These peptides can either stimulate or inhibit activity. Even with this very basic arrangement, the nerve net is remarkably effective. It coordinates essential behavior such as the contraction of tentacles during prey capture, the opening and closing of the mouth, digestion, and the rhythmic pulsing that allows a jellyfish to swim. This rudimentary nervous system represents the very first appearance of neurons in the animal kingdom and shows how even a scattered network of cells can produce coordinated responses. Most animals we will study later possess a complete digestive system with two separate openings. Food enters through a mouth and waste exits through an anus. Cnidarians, however, have a much simpler arrangement known as an incomplete digestive system. They have only one opening that serves both functions. This single opening is located at the center of the ring of tentacles and functions as both mouth and anus. Look at the image in the upper right. You'll see filled in a gray color the gastrovascular cavity, sometimes referred to as the coelenteron. This cavity allows for extracellular digestion after prey is pushed inside. The lining releases digestive enzymes that begin breaking down the food outside of the cells. Once reduced to small enough molecules, they are absorbed. And this contrasts with the poriferans that could only do intracellular digestion. Cnidarians have no specialized organs for gas exchange, circulation, or excretion. They rely entirely on diffusion for oxygen and carbon dioxide exchange. Due to the absence of a circulatory system, there is one reason for their bell-shaped medusae bodies, and that is relying on diffusion means they cannot be too thick, or else diffusion alone won't result in enough gas exchange, right? Um, so for those central regions, they just wouldn't get access to oxygen, and the cells wouldn't be able to survive, nor would they be able to dump out their waste like carbon dioxide as well. So having a non-living mesoglia allows them to get away with a thicker body since it is a gel-like matrix and not made of living tissue. The Cnidarians move us from the parazoa of poriferans to the eumetazoan clade, and this means that they must have tissue layers, and to be more specific, they are diploblastic. Here we can see the Cnidarians broken down into four major groups: the cubozoan, scyphozoa, anthozoa, and hydrozoa. Looking at them, you can see they all have prominent stinging tentacles and shown either in medusa or polyp form. Not shown in this tree is that all cnidarians fall under one of two monophyletic subphyla, the Anthozoa or Medusazoa. Now, we covered Medusazoa earlier when we looked at polymorphism. The subphylum Anthozoa includes sea anemones and all corals. One of the distinguishing traits is that these members stand out morphologically as only having the polyp body form throughout their entire life. Anthozoa never produce a medusa stage because they are permanently attached to the substrate. They are all sessile. The subphylum Medusazoa is larger and more varied. It includes three main classes: Scyphozoa, Cubozoa, and Hydrozoa. In most members of Medusazoa, the medusa stage is prominent, although the polyp stage is still present in the life cycle of many species. This subphylum includes the classes Scyphozoa and Cubozoa, which are mostly free-living, swimming medusa, and they forms commonly known as they have forms that are commonly known as jellies. In these groups, the polyp stage is usually short-lived or reduced, and the third class, Hydrozoa, is the most diverse. Most hydrozoans alternate between polyp and medusa stages during their life cycle. Some have only polyps, others have only medusae, and many others form colonies. So finally, we have the hydrozoa. Again, this group includes sessile forms as well as swimming colonial forms, such as one really interesting example called the Portuguese man of war, which we're going to look at in greater detail. These variations within Medusazoa show how the basic cnidarian body plan has been modified in different ways to suit different lifestyles within the same evolutionary lineage. Let's look at Anthozoa in greater detail. The name Anthozoa literally means flower animals for the beautiful floral-like appearance of the tentacles of in one of its members, the sea anemones. But don't forget that the corals are included within Anthozoa as well. Their bodies are usually cylindrical in shape. Even though the coral often have a branching pattern, they're still cylinder-like, each of the branches, and they are fixed to a substrate. Sea anemones have a lot underneath the hood, going on underneath the hood. Sea anemones give us the clearest picture of how an anthozoan polyp actually works. Around the central mouth, you see a ring of tentacles loaded with cnidocytes for both defense and prey capture. The mouth leads into a flattened pharynx. So here is the pharynx right here. And this is lined with ectoderm. and is actually biradially symmetrical. So if you imagine this pharynx has a specific structure to it that you can't just run a plane any direction you want down the whole length of the body. Only specific planes, two different planes, will make it symmetrical. That means it is biradially symmetrical. A special ciliated groove down the pharynx here is called the siphonoglyph, and this runs along one side of the pharynx, and it actively directs water inward to keep the cavity inflated and to bring in fresh oxygen. The gastrovascular cavity inside is divided by sheets of tissues called mesenteries. These mesenteries increase the surface of area available for nutrient absorption and also provide mechanical support so the body doesn't collapse on itself. Sea anemones feed on small fish and shrimp. They use their nematocysts to immobilize the prey, then pull it into the mouth with the tentacles. Many sea anemones also form mutualistic partnerships. Some live with hermit crabs, trading food for protection. Others host clownfish, which gain safety from predators while defending the anemone in return. And this relationship shows how even a simple polyp can participate in complex ecological interactions. A quick look at this video, you can see the sea anemone in motion. There's a fish at the bottom left at the beginning here, just for a moment, and it gets stung and paralyzed, and then the tentacles retract in a coordinated movement while a siphonoglyph keeps a steady current flowing water into the pharynx. As mentioned, they've evolved several symbiotic relationships. To the right, you can see clownfish moving freely among the tentacles without being stung. The fish is protected with a sea anemone's reach, while the fish help to chase away potential predators of the sea anemone and also contribute by sharing food scraps. This is a classic example of a mutualistic symbiotic relationship. In the video to the right, a crab has lifted a sea anemone from its substrate and relocated it to the top of its shell, providing a defense system from prey coming from above. I'm not quite so sure if this is mutualistic because it may or may not be beneficial to be hauled around in such a manner. So this may land under either commensalism, which would mean the crab benefits, but the sea anemone doesn't benefit and it's not harmed either. Or it could be parasitism, where the sea anemone is actually harmed in this process, but it has no other alternative. This brings us to the checkpoint question 4. What directs water into the pharynx of sea anemones? Sea anemones have both sexual and asexual options in their lifestyle, and neither will employ a medusa. During sexual reproduction, the planula larva stage is particularly interesting. In many species, the planula actually passes through a brief bilateral symmetry phase before it settles and transforms into the radial adult polyp. This temporary bilateral stage is a reminder that cnidarians are still early in animal evolution, and asexual reproduction is also common. As an anemone slowly crawls across a rock, small pieces of its petal disc, the foot-like base, can tear away or be deliberately left behind. Each fragment can regenerate into a completely new anemone. And this combination of sexual and asexual reproduction gives these sea anemones quite a bit of flexibility to colonize new areas. Corals belong to the same subphylum Anthozoa as sea anemones, so they also live their entire adult lives as polyps only. Remember, in Anthozoa, there's never a medusa stage. The big difference between corals and their Anthozoa is that most corals are colonial. Hundreds of thousands of tiny individual polyps live together in a single structure, sharing a common gastrovascular cavity. Each polyp is much smaller than a typical sea anemone, but it has the same basic body plan: a mouth surrounded by tentacles, a pharynx, and a gastrovascular cavity divided by mesenteries. What makes corals unique is that they secrete a hard external skeleton made of calcium carbonate. Over time, these skeletons build up into massive reef structures. Corals feed on planktonic organisms such as algae, bacteria, and larvae that drift past their tentacles. Because they are sessile, they depend on water currents to bring food to them. This colonial lifestyle with a calcium carbonate skeleton has allowed corals to create some of the largest biological structures on Earth and to support an enormous amount of marine diversity. The success of reef building corals depends on their amazing symbiotic partnership. Each coral polyp lives in a mutually beneficial relationship with microscopic algae called zooxanthellae. They live inside its gastrodermal cells. They provide fixed organic carbon to the coral, such as sugars, which support the coral's growth and survival. The algae have photosynthetic pigmentation, which gives corals their brilliant coloring patterns. And since this is mutualism, we have to consider the benefit to the algae as well. Just like we saw in lichens or within poriferans, the coral provide a cozy, protected environment within the coral polyp and access to the coral's waste, which the algae can recycle as a source of nutrients. Before shifting over to our next major clade, the Medusozoans, it's worth taking time pointing out how fragile this relationship is. Colonies of coral make up the foundation of many marine ecosystems we call coral reefs. These reefs are well known for having some of the highest levels of species richness, which makes their preservation in the interest of the species of humans. When corals become stressed by warmer water temperatures, pollution, or other environmental changes, they expel the zooxanthellae in a process known as coral bleaching. Without the algae, the coral loses its color and turns white because the white calcium carbonate skeletons show through the transparent tissue. And more importantly, the coral loses its main source of energy. If the stress is short lived, the algae can return and the coral can recover. But if the stress continues, the coral will starve and eventually die. This series of events can lead to collapse of the entire reef system. We now move to the polyp-only Anthozoa into the other major subphylum Medusozoa. Within Medusozoa, the class Scyphozoa contains the animals we commonly call true jellies. They are almost entirely marine. In this group, the Medusa stage is the dominant and most visible part of the life cycle. The polyp stage is usually small and short-lived. Scyphozoans have the classic bell-shaped body that most people picture when they think of a jellyfish. They range in size from just a few centimeters to more than 2 meters across in the largest species. These animals are free-swimming, solitary carnivores that drift or gently pulse through the water column. Their medusa form, their medusa form is perfectly shaped for a free-floating lifestyle. The bell shape and muscle rings allow them to move slowly but effectively while their long trailing tentacles capture prey. Look at the image to the left, or actually, sorry, to the right. It depicts the structure of a typical Scyphozoan medusa. There is an opening on the underside of the bell, serving as the mouth and anus, and is surrounded by hollow tentacles packed with nematocysts. The mouth opens into the gastrovascular cavity, which is divided into four interconnected sacs called diverticula. In many species, these sacs branch further into radial canals that spread throughout the bell. The reason? By breaking up the digestive system into several branches. This increases the surface area of the lining of the digestive system. And this is the region that is responsible for nutrient absorption and gas exchange. The gastrovascular cavity also helps support the body and distributes the digested food to all parts of the medusa. Because the animal is mostly water and has a thick mesoglea, it can achieve a large size without needing complex internal systems. And checkpoint question 5: what is the primary stage in the life cycle of Scyphozoa? The lifestyle of Scyphozoa shares many parallels to what we've previously discussed for Cnidaria, but with a few things we'll add. Scyphozoans possess a nerve net that encircles the edge of the bell and a nerve ring that helps coordinate swimming and feeding movements. While they do not have true muscle tissues, we've said before that diploblasts can have contractile cells. The Scyphozoa use a set of contractile cells that creates a muscle ring around the bell and provides contractile force that allows the animal to pulse and propel itself through the water. These jellies have separate sexes, with the gonads forming directly from their gastrodermis. That's the inner layer, if you'll recall. Fertilization occurs externally in the water, and the resulting zygote develops into a planula larva. The planula eventually settles onto a suitable surface and grows into a small polyp. That polyp then begins to bud off new medusae. In this way, the life cycle alternates between a sexual medusa stage that produces gametes and an asexual polyp stage that allows the population to grow and persist locally. One other side note is that the scyphozoans have sensory organs called rhopalia, which may be found in pockets along the bell's edge that can sense orientation, light, or touch. The ring of muscles around the bell, as shown in this image, allows the bell to contract, and that pushes water out and creates a jet that propels the jellyfish forward. So, with a Scyphozoa, no fins or other specialized swimming structures are necessary. Checkpoint question 6: what does the life cycle of most Scyphozoans involve? We now turn to the class Cubozoa, commonly known as box jellies. So these aren't true jellies. Cubozoa, we're talking about box jellies. These animals have a distinctive body shaped medusa that is usually small, only ranging from 15 to 25 centimeters. So there's 2.54 centimeters in an inch. So we're looking at maybe up to about 10 inches in size. At each corner of the box-like bell, you will find muscular pads called pedalia, and from each pedalium, one or more tentacles extend from it. Cubozoans are famous for having the most advanced eyes of any cnidarian. They possess image-forming eyes, complete with a cornea, lens, and retina. These eyes are arranged in four clusters, one between each pair of pedalia. This gives the box jelly remarkable vision for an animal without a brain. They are active hunters. They can swim through the water column searching for prey such as small fish, worms, and crustaceans. Like other medusozoans, they have separate sexes, and the planula larva settles to form a polyp, producing new medusae. We now come to the last major class within the subphylum Medusazoa, the hydrozoa. Hydrozoa are extremely diverse and include nearly 3,200 species, and most hydrozoans are marine, but a few live in fresh water. We covered a member of this class, the Obelia, in our explanation of polymorphism found in Cnidaria. However, there is a lot of variation. For example, the well-known Hydra has only the polyp form throughout its life. Other hydrozoans have small medusa stage called a hydromedusa. Hydrozoans show a wide range of body forms and lifestyles. Some are solitary, while many form complex colonies of specialized polyps. This diversity makes hydrozoa one of the most interesting groups to study when we want to look to see how basic cnidarian body plans can be modified in many different ways. One of the most famous and dramatic members of the hydrozoa is the Portuguese man of war, Physalia physalis. It received its name due to the comparison to the Portuguese naval warship man of war. Although it looks like a single large jellyfish, it is actually not one. It's not one individual at all. It is a free-floating colony made up of many specialized individuals called zooids that work together as a single unit. The most visible part is the gas-filled float on top. This is known as a pneumatophore. The structure acts like a cell and keeps the colony at the surface of the water. Hanging down from the float are long tentacles and other specialized zooids that perform different functions for the colony. The Portuguese man of war is divided into four main types of zooids, each with a specific role. The pneumatophore is the gas-filled float that provides buoyancy and keeps the colony at the surface. The dactylozooids are the long tentacle arms with powerful nematocysts. These are responsible for capturing prey by stinging and paralyzing fish and other small animals. The gastrozooids are the digestive members of the colony. They engulf and break down the captured prey and distribute the nutrients to all the other zooids. Finally, the gonozooids, which are the reproductive members. They produce sperm and eggs for sexual reproduction. All of these different zooids are genetically identical and they work together as one functional unit. This level of specialization within a colony is one of the most advanced forms of organization found in hydrozoa. So we're going to just run through this really quickly here, but the Portuguese man of war, again, it belongs to the hydrozoans and specifically a group called the siphonophores, and they're free. These are free-floating colonial organisms. Again, they're made up of many specialized medusoid and polyp individuals that all work together. The colony, again, it's not a single animal, but a group of many interdependent zooids. And that gas that you find in the pneumatophore, interestingly enough, it's not CO2, which is a common gas that's released through cellular respiration. It's actually carbon monoxide. And that's what gives the hydrozoan in this example that a blue bottle name. It looks like a blue bottle floating at the surface. The entire structure is actually carnivorous and they feed on soft-bodied marine animals by using their tentacles, which can stretch many meters. And of course, they're covered with stinging cells used to capture those prey. Because the different zooids perform different tasks, the colony functions almost like a single complex organism. And this is one of those most impressive examples of colonial organization in the animal kingdom. Checkpoint question 7 asks, what is the role of dactylozooids in a colony? All right, well, to wrap up our look at Hydrozoa, it is important to remember how diverse this class really is. Some species, like the Portuguese man o' war, form huge free floating colonies with highly specialized zooids. Others, such as Hydra, are simple solitary polyps. Some hydrozoans have both polyp and medusa stages, while others have lost one stage entirely. Another consistent feature across the group is that their gonads develop from epidermal tissue rather than gastrodermal tissue. This small detail helps biologists distinguish hydrozoans from other cnidarian classes. Overall, the hydrozoa demonstrates an incredible versatility of the basic cnidarian body plan and shows how colonial living has allowed these animals to evolve complex behaviors and structures while remaining diploblastic. And that actually concludes our survey of Porifera and Cnidaria. We saw how sponges represent the earliest and simplest multicellular animal organization, and how cnidarians took the next major step by evolving true tissues, radial symmetry, and a more sophisticated body plan with two distinct forms. I hope you found this presentation enjoyable, and I hope to see you in the next one as we continue our exploration of Animalia.